All right, thanks, Hagen, for that very kind introduction. It's great to be here. It's been really fun so far. Looking forward to the rest of this workshop. So today I want to talk about some of our recent work on using interpretable models to understand splicing. In general, our lab is interested in both splicing and some other aspects of RNA biology, including characterizing RNA binding proteins, and the connections between splicing and other steps in gene expression. So the main theme today is this new CatMap algorithm, which is basically a way of making a new type of RNA maps, which we call RNA activity maps, to describe the regulatory activity of splicing factors. And it does not use CLIP, unlike other types of maps. And it's based on an explicit biophysical model of binding and activity, and it can be used to infer which exons are direct versus indirect regulatory targets, for example, and even to figure out which splicing factors may act synergistically with one another. We recently posted it to bioRxiv. So generally, when we think about the activities of splicing factors, we think about SR proteins as binding to exonic splicing enhancers and recruiting or stabilizing the interactions of SNRPs with adjacent splice sites. And we may also think about H&RP proteins, which often bind exonic splicing silencers and somehow inhibit recruitment of the splicing machinery to exons. But of course, there are other types of activities for more alternative splicing regulatory factors. So traditionally, the way that we describe the activity of a factor, and this is due originally to Bob Darnell, is one looks at a data set, say an RNA-seq data set, with a control condition and a knockdown or overexpression condition or a knockout mouse or some sort of perturbation of the factor. And one identifies exons that are, say, upregulated after knockdown, in which case one would infer that the factor repressed the splicing of that exon, perhaps. And you look at where the factor bound, typically by CLIP, and use that to make inferences about sort of which locations of binding are associated with which types of activity. And so when you apply this here for the factor PTB, and one looks at the exons that are upregulated following PTB knockdown, this is data from ENCODE, you see that the upregulated exons tend to have CLIP peaks either in the exon or upstream. And the way that the RNA map is made is simply by adding up the CLIP density across all those exons that are upregulated. That gives you this red curve here. And all similarly for those that are downregulated here, PTB doesn't look like it has any positive activity. And comparing to the unchanged as sort of a control. And then the summary here is that PTB represses when it binds upstream or in exons. So when Michael McGurk, postdoc, joined the lab, and I told him about, this is how RNA maps work, he was like, that really bothered him. He felt that this could not be the way that we should describe splicing factors. It's based on CLIP, which is notoriously not a very quantitative measure, and we're simply just adding it up, and there's no underlying model of the regulatory activity. It's sort of describing where the binding is rather than kind of modeling what the activity is. And so I was like, okay, try to come up with something better. So he thought about that for a while. And after a couple of years, it turned out to be a harder problem than we thought. He developed CatMap, which uses in vitro binding data. So describing what kinds of motifs the protein likes to bind, and then knockdown RNA-seq data. So I just want to tell you about that. So basically what he's trying to do is identify locations near exons where the factor can influence the splicing of those exons, and then to identify which exons are direct regulatory targets of the RBP. And it turns out that in some cases, we can also use the model for other purposes. And one of those can be to identify candidate secondary regulators. So splicing factors, when you knock them down, there's often some sort of compensatory regulation or other factors change, and those can influence some of the splicing factors, some of the exons detected in RNA-seq experiments. So we want to be able to infer those as well. All right, so imagine you have a splicing factor. It activates when it binds in the downstream intron. It represses when it binds to the upstream intron and does nothing when it binds the exon, let's say. We take some RNA-seq data from control conditions and knockdown conditions. We have a model of the binding motif, and this is required. And we like to use biophysical models such as position-specific affinity matrices that can actually output a predicted affinity for any given k-mer. But we can also use weight matrices, for example, from RNA compete or from CLEX. You just need some kind of reasonable model for the motifs that are bound by this factor. And then the sort of this binding model, which I'll describe in more detail, kind of goes through an activity map. And so here we have parameters that describe the activity of the factor, for example, enhancing, promoting exon inclusion as a function of distance from the splice junctions. So positive is enhancing, negative is repressing. And then one kind of applies this across all the exons to infer whether the exon is going to be changed or not. And then this model here, we sort of start with an uninformative prior, and then we learn what's the activity map that best explains the data. So the structure of the model is a generalized additive model. So we are trying to predict the log odds that a given exon was downregulated following knockdown, for example. And that is modeled in terms of a binding function here that relates the essentially calculates the occupancy or the change in occupancy of the splicing factor in the vicinity, in a particular region adjacent to an exon as a function of the free protein concentration and then the change in free protein concentration that happened following knockdown. Both are parameters that we learn. And this change in binding is then multiplied by this alpha J, which is the activity of the factor at that particular location relative to exons. And so that's really the quantity that we want to learn. And then importantly, in this model, Michael added these other terms, these linear terms, which reflect other features which can influence the likelihood that you'll detect significant regulation by a program such as RMATs, or one could use MAGIC as well. And these are features like the expression level of the gene, higher expressed genes, give you more reads, make it easier to detect changes in splicing. Also the initial psi value of the exon. If it's starting at 98% inclusion, it's much easier to detect downregulation than upregulation, for example. So that's what those terms are. And so for the binding models, we either use data from our lab's RNA bind and seq, or we can, as I mentioned, we can also use motifs from to the Tim Hughes lab RNA compete. And what we do is we start with an exon. We consider the regions shown here, plus or minus 200 into the introns and 60 bases into the exon relative to the splice junctions. And we have one or more matrices that describe the binding affinity of the protein, and we kind of slide those along these regions and add them up and get an RBP score, which is kind of a relative affinity, where here, like 0 would be like a perfect binding site, and the negatives are sort of the log affinity relative to that. So one does that with a set of exons that were measured in an RNA-seq experiment. You get to sort of these RBP binding scores. And then we have a model for how occupancy depends on affinity. And so that's sort of a sigmoid function that depends, again, on the free protein concentration, which will be higher before knockdown than after knockdown. And so really we assume that it's the change in occupancy that dictates whether there's going to be a change in regulation. And so we take the difference of those sigmoids, gives you a curve like this. And then we describe this particular the bottom here. This is the so-called most relevant affinity. This is the affinity where you'll get the biggest change in occupancy following the knockdown. And so then we convert these affinities to then changes in binding, and then we multiply those changes in binding by the activity map, and that gives you what we call here the RBP impact. And so that's the product of the binding change at each position by the activity at that position, and then you can sum that to get a prediction of the log odds of whether that exon will actually change, and then we compare that, of course, to the actual change. And so, importantly, this quantity here, this RBP impact, is a sort of exon-specific quantity that tells you how likely this factor is to directly regulate that particular exon. And it is a fully Bayesian approach. So we have a few hyperparameters, and we have sort of very uninformative priors on them, and then we can kind of do a full posterior sample. So for example, we don't know the free protein concentration, and we don't even necessarily believe that we have accurate absolute affinity measures. And so we kind of learn this most relevant affinity parameter kind of considering different versions. So for example, in this case here, we assume that the protein was at moderate free protein concentration. So it's sort of the higher affinity sites that will lose the most occupancy. Whereas here, we assume that it's kind of proteins present at saturating levels, so that really high affinity sites don't actually, the protein doesn't fall off them with the knockdown. It's only the moderate affinity sites. And then you get, a very different landscape of binding changes. So we consider that as well. And same, also with the other parameters. So we assume that the activity of the factor at each position can be anything, but that the change in activity is kind of smooth. That is, that if you have a strong positive activity at position plus 30, that at position plus 40 is probably also going to be positive. So there's a Gaussian smoothing parameter there. But that the activity can change when you cross this, cross from exon to intron, of course. And so then we just average over all the possible values of the parameters and get a posterior change in binding, for example, posterior most relevant affinity value, as well as posteriors on the activity parameters. And so here you can see that we're very confident that this factor activates in the downstream intron, for example, and fairly confident that it represses in the upstream intron. And then we don't really know what's going on, if there's any activity in the exon. So Michael did a large number of simulations here where he took sample activity maps and then simulated RNA-seq data, and then asked, could we infer the activity map back, that we had used. And he found that generally we could, and then also that the probability of regulation of a given exon predicted by the model was well calibrated with the actual likelihood that it would change. Again, using kind of held out portions of the data. And when we apply it to real data, so here, of course, RB Fox, well understood factor, we found that we get a very sensible map with sort of high confident activity in the downstream intron, positive activity, and sort of a moderate repressive activity in the upstream intron, and then sort of weaker repressive activity in the exon, which is similar to the map you would get from CLIP in this case. Here are some examples of RNA maps or activity maps for other well-known factors. So PUF60, a homolog of U2AF, activates when it binds very close to the 3' splice site. HNRPC represses in both flanking introns. K represses mostly in the upstream intron and the exon. And TRA2AB is a SR-related protein that activates in exons. And we find that the activity map that you get from, say, two different RNA-seq replicates is highly correlated. And it's also highly correlated when you compare, for example, the activity map from K562 cells versus HEPD2 cells and from the ENCODE data, or, for example, mouse knockout data versus the human factor is correlated as well. As you might expect, the factor has sort of an intrinsic activity of how it affects splicing. That is independent of context. And of course, we wanted to then, we hadn't used CLIP in the derivation of these models, so we wanted to use it sort of in testing. And so here we compared, for example, for quaking QKI, this is the map we get with strong positive activity downstream, weak repressing activity upstream. And we see support for that in the CLIP. Although you'll notice here that we infer repressive activity in the intron, but no repressive activity in the exon, whereas the CLIP signal for suppressing targets extends into the exon. And so here's an example where we make a very distinct prediction here that we think that this is just CLIP signal like bleeding into the exon from peaks in the intron, and that actually it doesn't have any activity in exons. Overall, we find we get strong peak enrichment in both suppressed and enhanced targets for almost all of the factors that we've studied when there's CLIP data available, as expected. Okay, so we also tested some of the predictions here using mini-gene splicing reporter assays, and so these were led by postdoc David McWaters, a Canadian. And what he did was to take predicted exons from several different factors, and then to look at the what point mutation can I make that will is predicted to strongly disrupt binding of the factor at a particular location. So for example, here's an exon predicted to be regulated by quaking. If he mutates this site here, it should lose regulation. And then he makes both the wild type and mutant mini gene, and then tests those with and without knockdown of quaking in cultured cells. And so he did this for several exons with several different factors, and the blue arrows here show you the expected direction of regulation, and in all but one case, the observed direction matched the expected direction. And the one case here was for PCBP1, where it has we mutated a pyrimidine-rich element that was close to the polypyrimidine tract and accidentally sort of strengthened the 3' splice site. So I think that's why that might have gone in that direction. But overall, it worked well. So when we apply CatMap to 20-something factors with available knockdown RNA-seq and binding-seq data from ENCODE, we get a wide range of different activity maps here. You can see sort of the classic SR activity, positive activity in the exon, not much in the intron. There's an H&RP pattern here where you tend to get repression in the exon, and then a variety of other types of activities, at least roughly half a dozen distinct types of activities. So I mentioned before that we can infer direct regulatory targets. And so we do a statistical test where you look at the model for a given exon and ask whether the model with the learned activity coefficients better predicts that particular exon and significantly predicts it significantly better than a simplified model with no splicing regulatory activity. And that gives us sort of a confidence that the exon is direct or indirect. And when we do this across all of the ENCODE data, what we notice is that most of the changes are indirect. Okay, so for example, first one, HRPK, it's a repressor, and so about 25% of the significant exons are predicted to be direct repression targets of K. But then there's also some exons that go up, that go down, and K doesn't have any activating activity, and so these are all indirect. Plus, we think many of these other upregulated targets are also direct, and so and that's typical here. And so, if you remember, the ENCODE data was generated by lenti shRNA knockdown followed by antibiotic selection for 5 days. Okay, so what we think has happened is that, and this is typical of splicing factor knockdowns, that there's lots, cells do not like to have splicing factors depleted. They respond, they upregulate other factors, and so forth. and so that there's a lot of indirect things that happen here. Okay. All right, so another experiment we did to try to understand indirect regulation and kind of test the model further was to basically look at all the splicing changes in a given experiment and then kind of make a meta model where we give it all of the models of all the splicing factors that we've learned to date and ask it to then predict which splicing factors changed in that experiment and by how much. And that's kind of what's shown here is the coefficient that the factor interprets, where 1 means, it's pretty sure it was knocked down, and -1 means it's pretty sure it was like overexpressed equivalently and so forth. And so what we find is that we can, the highest coefficient is almost always the factor that was actually knocked down in these experiments. And so we can, and we did this in a way where it's not, where we're kind of giving it the data from one cell type and asking it to predict from a different cell type, so it's held out data. And so that's reassuring that we can infer the knockdown factor. But you see there are some off-diagonal terms here. For example, in the H&RPC knockdown of K562 cells, there's a significant positive effect of PCBP1, suggesting that PCBP1 might be exerting some secondary changes on splicing that would explain some of these indirect effects. And we noticed actually that PCBP1 changes in many of the ENCODE knockdowns. And when we group the knockdown experiments by whether the PCBP1 mRNA was significantly downregulated, upregulated, or unchanged, we found that the estimated coefficient for PCBP1 was strongly related to that in the correct direction. So suggesting that PCBP1 is sort of densely connected in the splicing regulatory network and is often exerting sort of secondary effects in many of these ENCODE experiments that we can then help to interpret. Okay. We also noticed that when CapMap is trained on the significantly changed exons, but you can apply it to all exons, this splicing impact. And so when we looked at the splicing impacts for the non-significant exons, and we looked at what direction did they change, we found that again the non-significant exons did change in the expected direction. So essentially, we can identify evidence of direct regulation even for things that were non-significant in an RNA-seq experiment. Okay, and then finally, I want to tell you about sort of a later sort of a fortuitous observation that Michael made. So Michael was looking at the different RNA maps and noticed that, as I mentioned, RB-Fox and quaking have very similar patterns with activation in the downstream intron, repression in the upstream intron. And when he was looking at the, thinking about, maybe they work together to regulate splicing, and what would that look like? And so the idea would be that if they did work together on a subset of exons, that if you looked at the exons that were downregulated following depletion of one of these factors, that you might see evidence for binding of the other factor in those exons. And just as a reminder, RB-Fox binds sort of a canonical UGC-AUG motif, and quaking binds a very different motif with consensus ACU-AAC. And these are both really important genes in the nervous system. And so this is what he saw. So when he looked at quaking targets where quaking had been depleted in human cells and looked at the RB-Fox impact of those exons, he found that the downregulated exons had significant negative RB-Fox impacts. And when he looked at exons that changed following RB-Fox depletion, he found significant negative quaking impacts, both in human cells and also in triple knockout mouse cells from Xiaolin Zhang's lab. So this suggests that they do actually co-regulate exons. And then when we looked at predicted binding motifs of RB-Fox and quaking in quaking downregulated targets, we saw lots of RB-Fox motifs near the quaking motifs. And we noticed that they tended to be very close by to the quaking motifs. And so we kind of measured the distances in the sort of co-regulated exons and found that they tend to be very close, about 25 nucleotides apart. closer than you would see in exons that didn't change in these knockdowns. And so we wondered whether the set of quaking FOX co-regulated exons might have some sort of functional relevance. And we did see a significant gene ontology signal for actin binding and cytoskeletal functions, generally, many of which are important in neuronal differentiation. And so if you're familiar with the expression of RBFOX and quaking, RBFOX is expressed highly in neurons, particularly goes up a lot during neuronal development, whereas quaking is prominent in glia and actually very low in neurons. So what we think may be going on here is that in neural progenitor cells or other sort of early cells in neuronal differentiation, you have significant levels of RBFOX and quaking, and there they act cooperatively to regulate the splicing of these exons here. And then as these cells differentiate either into neurons or various types of glia, either RBFOX goes up and quaking goes down, or quaking goes up and RBFOX goes down, and you would sort of lose the co-regulated exons and get just a different program that's specifically regulated by that specific factor. Okay. So in conclusion, CatMap infers splicing activity patterns from just binding and knockdown data. It seems to generalize pretty well across cell types and species, allowing you to skip doing a lot of clip experiments. We can infer which splicing factor was perturbed from a large set of knockdown experiments. We can identify direct regulatory targets, including some that are not significant, by sort of a regulatory splicing regulation detection algorithm. And sometimes we can identify secondarily impacted splicing factors. And in this case, we sort of are able to detect a potential synergistic relationship between RBFOX and quaking. And so I'm just going to thank the people, and then I have a few more slides after this. So this is my lab on Canadian Tuxedo Day. I have several Canadians in the lab, 3 or 4 at last count, and a lot of Canadian spirit. And Michael led the sort of computational analysis in this study, and David did all the experiments. And I want to thank our funders and collaborators as well. And then I think I have a couple more minutes, so I'm just going to briefly talk about another topic just to sort of provoke some discussion. And so in the past few years, the problem of sort of predicting splicing like de novo, like from the primary transcript, predict where all the splice sites are or what the canonical splicing pattern will be, has been addressed, and there's been some really important progress. And I want to sort of talk about 3 different types of models today. So black box models, where we really don't understand how they work, but maybe extremely accurate. White box models, where we have a really, there's an explicit model of how splicing works that's encapsulated in the model that we can sort of easily pull out. And then gray box models, which are sort of in between, and what roles these kind of models may play in various types of analyses. So many of you are familiar with Splice AI, which came from Kyle Farr's work at Illumina. And this is a model that uses a very deep neural net to predict from human genomic sequence whether any given position is a 5' or 3' splice site. And it actually uses a very wide window of up to 10 kb, or like 5 kb on each side of the splice site to get its most accurate predictions. It's highly accurate, greater than 90% in both human and mouse. And it can also be used to predict the splicing phenotypes of genetic variants. However, it is a black box method. It's like a 32-layer neural net, and it's like really impossible to get anything from the parameters in the model. And even from in silico mutagenesis, it's very hard to figure out what it's doing. We've done, with our collaborators, some fairly extensive in silico mutagenesis, and we don't understand. Splice AI knows about 5', 3' splice site motifs and so forth, but it doesn't seem to be using the canonical splicing regulatory elements that we would be expecting it to use, it's using some other information that is still sort of mysterious. Okay, so with some collaborators, Armando Solar-Luzama in computer science at MIT and Phil Sharp, we developed recently we call a gray box model. And so this is a model where we want to use the power of neural nets, but we don't want to let it just learn information that's very far away from the splice sites, which typically we think is not used by splicing machinery. So what we give the model is not the raw sequence, but it's a pre-processed version of that sequence. So first we scan it for potential splice site motifs and potential binding sites of known splicing regulatory elements, and we say, here's where all the splice sites are. Here's a score for 5' and 3' splice sites at each position, and here's a score for RBPs, say from RNA binding seq, affinities. Here's just the highest affinity sites, and then we give that to the model, and we say, learn how to predict splicing from that. And so this was recently published, so you can read all about that. The main conclusion is that it's not quite as accurate as Splice AI, but it is more interpretable. It can tell you which factors regulate a given exon, and we actually learned some new motif models for some of these factors that seem to work better for the prediction task and work comparably well for the initial sort of analysis of the binding seq data. So they may reflect sort of slight differences in binding that happen in vivo versus in vitro. And then finally, I want to mention some recent work from Kayla, a former grad student in the lab who recently graduated, also published. And in this model, so it's a semi-Markov model, very similar in spirit to GenScan, but it only uses information that the splicing machinery might use. So it uses kind of maxent models of the splice sites, and then it learns regulatory activity of different 6-mers and kind of puts those all together. And it's only about 70% accurate in mammals, but very accurate in virtually all other organisms that we've tested, including about 85% accurate in zebrafish, Drosophila, Arabidopsis, and so forth. And the motifs it learns, if you take like the 6-mers and their scores and kind of cluster them, you get motifs that strongly resemble known regulatory motifs. So for example, a G-rich motif that looks like HRBH and F binding sites, you get SR-like motifs that are activating it, and so forth. And so it has a number of applications in terms of, we think we can sort of infer what are the main positive and negative acting regulatory, splicing regulatory motifs in an organism. Okay, and I'll just end there, and thanks for your attention.
Thanks a lot. That was really great. One quick thing. I don't know if you saw this paper by Tim Hughes, but they looked at Splice AI, and Splice AI works really well for predicting splice sites for protein coding genes, but not non-coding genes. So maybe it knows something about NMD.
Yeah. There's it might know something about reading frames. Right. Might have picked that up, the periodicity, and so forth, and so it might be using that information. Yeah, that's certainly possible. That's a good observation. Yeah.
The other thing is we see quaking by bipartite motifs a lot. Mm-hmm. And you probably saw the same thing in RNA bindings. I can't remember if that's in your paper. And do you think that's related to this relationship with RP-Fox?
That's a good point. I mean, our model will, if it's the two halves for quaking are similar to one another, I believe, and so our model will kind of scan its quaking model, and those will kind of add up and be interpreted as a stronger quaking motif, but it's not like explicitly modeled. But it would be interesting to look at the bipartite ones versus the monopartite ones and see whether one of them interacts with Fox more or something. That's a good. Thanks.
Fox site that downstream of the quaking site?
Not always. I think there's a tendency, but not always. Yeah.
Okay, all right, thanks.
Just generally, people at home can't hear unless you ask the question to the microphone.
Chris, amazing talk. So, two quick questions. So, splicing factors, many of them are tissue specific. Is CatMap able to determine tissue specificity, or, for instance, RB-Fox, or quaking has a nuclear isoform, a cytoplasmic isoform, any of those variations? And the other question is, have you looked at pseudoexons or exon-containing alus, or any cryptic exons, and see what comes up?
Yeah, so a lot of questions there. Let me just say about the ALU part. So HNRPC, I think Gil Ass published that HNRPC represses a lot of ALU exons, and we see that very strongly in our data that like about half of all the ALU exons, are predicted to be direct regulatory targets of HNRPC. Many change in that knockdown. Regarding tissue specificity, we've been keeping to primarily ENCODE data, so we're limited a little bit there to factors that are expressed in K562 and HepG2 cells. But we've tried a little bit with some knockout data and so forth. The main limitation here is, you got to process the RNA-seq data and so forth and call the splicing changes. And so, but in principle, I mean, the software is available. And if you have some RNA-seq data you're interested in where the factor is perturbed, you can like learn your own models. That please, access software, try it out for yourself. Yeah.
Okay, thanks.
Hi. I'm interested in a lot of sex dimorphism responses in the immune system. And some of that comes from alternative splicing that seems to be hormonally regulated, but I think it's probably not well understood all of the factors that contribute to different isoforms being expressed in different sexes. So if I am interested in understanding sex dimorphism, which of these models do you think would be most apt to capture that those mechanisms?
Yeah, I would suggest that CatMap could be useful here. So when you run it in this mode where you give it like all the models of all the splicing factors that we know, then you can give it two different RNA-seq sets. For example, immune cells from different sexes or something, and you can ask it like, which splicing factors do you think are perturbed between these two sets, and it will give you some inferences about this one is up and that one's down, and so forth. And so that's an application which we're starting to explore, and we'd be happy to, if you email me or Michael, we'd be happy to sort of help you do that.
Yeah. All right. Thank you.
Okay. Thanks.